Double fertilization. Fertilization can be defined as a fusion of male gamete with a female gamete to form a diploid zygote. Fertilization in angiosperms is unique. It is known as double fertilization. This is because there are two sperms per pollen tube. One of the sperm fertilizes the egg cell to form a zygote, while the remaining fertilizes with the polar nuclei, resulting in formation of a triploid endosperm cell. The phenomenon of fertilization was first reported by Strasburger in Monotropa. The male gametes are brought to the egg present in the female gametophyte by a pollen tube. Fertilization involves the following steps: germination of pollen and the pollen tube, entry of the pollen tube into the ovule, entry of the pollen tube into embryo sac, discharge of the pollen tube contents, and double fertilization. Coming to the first one, germination of the pollen and the pollen tube. Siphonogamy, the delivery of non-motile sperm to the egg via pollen tube, was a key innovation that allowed flowering plants, that is angiosperms, to carry out sexual reproduction on land without the need for water. This process begins with a pollen grain alighting on and adhering to the stigma of a flower. The events and interaction that occur during the pre-zygotic cellular and molecular courtship between the haploid pollen and diploid gynoecium has been termed the pollen pistil interaction. The pollen capture and adhesion, pollen hydration, germination of pollen to produce pollen tube, penetration of the stigma by the pollen tube, entry of the pollen tube into the ovule, and discharge of the sperm cells. Coming to the first one, that is pollen capture and adhesion. Angiosperm stigma can be classified into two types, wet and dry, depending upon whether or not they possess a surface secretion. Although there are examples of intermediate semi-dry stigma also. Secretions on wet stigma can be primarily lipidic, containing complex mixture of long chain saturated, unsaturated triglycerides, or primarily aqueous and carbohydrate rich. Stigmatic secretion play an important role in pollen capture and adhesion. On making contact with the wet stigma, pollen is quickly trapped by the surface tension and immersed within it. This first phase of pollen stigma interaction appears to be passive and indiscriminate process. Coming to the second one, pollen hydration, that is activating metabolism. After successful adhesion of pollen, the stigma can hydrate a compatible grain while restricting the hydration of foreign or incompatible pollen on the same papillus. Hydration transforms a pollen grain from a non-polar cell to a hydropolarized cell. Within minutes of hydration, pollen organizes its cytoplasm and cytoskeleton structures to support extension of single tube. Third one, germination of the pollen to produce a pollen tube. The pollen grains, by absorption of liquid from the moist surface of the stigma and protrusion of intine through a germ pore. A small tubular structure which arises in this way then continues to elongate, making its way down the tissue of the style and stigma. Almost all pollen grains are monosiphonous, that is, each pollen grain produces only one pollen tube. Other pollens, such as that of some species of families Malvaceae, Cucurbitaceae, are polysiphonous. Third one, germination of pollen to produce a pollen tube. The growth of pollen tube of a flowering plants is restricted exclusively at the apices. Microscopic examination of growing pollen tube reveals that most of the cytoplasm is restricted to the apical region, while large vacuole fills the grain and the older regions of the tube. The cytoplasm is restricted to the apical region of the growing tube by the formation of series of callose plugs at regular intervals behind the tube. Next, penetration of the stigma by the pollen. The contents of the pollen then move into this tube. The tube nucleus, the pollen tube grows through the tissues of stigma and style by secreting the enzyme to digest them, and it makes its way between the stigmatic papillae into the tissues of style. Next, penetration of the style by the pollen tube. In relation to transmitting function and anatomy, styles can be classified into three different types: open, closed, and semi-closed. The open style is characterized by a canal lined with a glandular epidermis or epithelium and is more common among the monocots. The closed style, usually present in edicots, has core of transmitting tissue. In semi-closed style, the transmitting tissue is limiting the stylar canal. Open type. Here, it is characterized by a canal lined with glandular epidermis or secretory epithelium. It is also called hollow style. The presence of epithelial and subepithelial cells with cytoplasm rich in organelles of the canal could favor the growth of pollen tube both through the cytoplasm of these cells and the canal, and the pollen tubes also seen grow through the thickened middle lamella between the epithelial and subepithelial cells, which it would probably the most favorable medium. Closed type. In closed type, usually present in edicots, has a core of transmitting tissue. Pollen tubes can grow intercellularly through the outer part of the cell membrane or between the plasma membrane and the cell wall. It is also called solid type. It is not known how pollen tubes penetrate and pass through the cell wall. One probability is the presence of enzyme that degrade cell walls. As pollen tubes grow within cell walls, the cytoplasm of the penetrated cells slowly degenerate. Semi-closed type. The transmitting tissue semi-closed style is characterized by production of a fluid intercellular matrix between its cells, and pollen tube generally grows through the highly specialized matrix. Entry of the pollen tube into the ovule and discharge of this pollen. After arriving in the ovary, the pollen tube finds its way through the style and enters the ovule. Depending on the place, the pollen tube in the ovule can be of three types: porogamy, mesogamy, selachogamy. Porogamy is the most common type. Here the pollen tube enters the ovule through the micropyle. According to some workers, porogamy is directed by filiform apparatus. Selachogamy. In this case, the pollen tube enters the ovule from the selagial end. However, the entry into the pollen sac occurs through the micropyle. Example, Casuarina. Mesogamy. In this, the pollen tube enters the ovule through the funicle or integuments. However, the final entry into the embryo sac occurs through the micropyle. But the pollen tube with two male gametes enter into the embryo sac only through the micropyle. The summary. Fertilization can be defined as a fusion of male gamete with the female gametes to form a diploid zygote. And fertilization in angiosperms is unique. It is known as double fertilization. The interaction that occurs between haploid pollen and diploid gynecium has been termed the pollen pistil interaction and consists of the style is of three types: open type, closed, and semi-closed. Depending upon the place of entry of the pollen tube into the ovule, it can be classified into three: porogamy, mesogamy and ctelagogamy. Thank you.